Hello, my name is Rebecca Smith, and I'm here to talk to you about predicting and preventing vector-borne diseases in Illinois. This is work that I came to somewhat late in my career, after I came to Illinois and started working with a colleague who was already working in vector-borne disease, but it's become a fascination for me and something that really does impact human health and animal health. And that's something I really believe in. I am a firm practitioner of One Health, the idea that we can't just artificially separate human, animal, and environmental health. And vector-borne disease is a classic One Health situation. So first, we should probably talk about what is a vector? So when we say vector-borne disease, what do we actually mean by a vector? Now, when we talk in epidemiology about disease, we often talk about the epidemiologic triangle, which is this idea of the host, the agent, and the environment combining to form the conditions for disease spread. In the case of vector-borne disease, there is some sort of biological agent. Usually we're talking about arthropods, insects, so mosquitoes, ticks, that live in the environment, that carry the agent, and that infect the host and spread the infection from host to host. So a vector is something that's alive, that moves around and carries this infectious agent from host to host, and it lives in the environment. This is why I say it's a perfect One Health scenario, because all of the aspects are there. We have wildlife, we have humans, we have the environment, all of those play a role in vector-borne disease. So let's talk about the vectors that we have in Illinois, and I'm going to focus on mosquitoes and ticks. And first, I should just talk about the basics of mosquitoes. And mosquitoes have a life cycle that we all may be a little bit familiar with, but basically an adult mosquito will lay eggs on a water surface. Those eggs hatch and you get larvae and pupa, which are actually aquatic. They live in the water. They can't live on land. Those pupa, the adult emerges from the pupa and becomes the airborne mosquito that we all love to hate. And then those adults then have to feed, mate, and then lay eggs to start the cycle again. Now, fun fact, my 8-year-old loves this fact, only the females bite. So the females bite so that they have enough energy to lay the eggs. The males are actually feeding on plant nectar. And all of these different aspects of the mosquito life cycle will play a role later when we start to talk about prediction and prevention. Because you have the aquatic phase, you have the aerial phase, you have the need to bite. All of that is part of the mosquito life cycle, and that's how we can think about where we can predict the mosquitoes will be and where we can control them. So in Illinois, we have two main vector mosquito groups. One that is an actual vector mosquito, and that's the Culex group. And there are multiple species in the Culex group. We have pipiens and restuans, a few different species, pipiens and restuans being the biggest ones. Those are these mosquitoes here on the left. You can see they're pretty little things. They're small. They're very common, and they live in floodwater. They like areas that flood and then dry up and flood and then dry up. They carry West Nile virus, which we'll talk about in a little bit. Now, there's another mosquito that we're worried about in Illinois, and that's called Aedes albopictus. You also might hear it called the tiger mosquito, or we're trying to get away from calling it the Asian tiger mosquito. And you'll see from this picture why we call it the tiger mosquito, because it's striped. It's beautiful, really. But you hate them. They like to live around people. They can lay their eggs in a tablespoon of water. and there's plenty of spaces with just a little bit of water around houses. They bite during the day, whereas the Culex mosquitoes prefer to bite at dawn and dusk. The Aedes albopictus mosquitoes in Illinois do not actually currently vector any diseases, but they can. We just don't have those diseases in Illinois right now, luckily, because the Aedes albopictus would be able to spread yellow fever, dengue, Zika, all of those diseases that we hear about from other areas. They could spread them in Illinois if those diseases existed in Illinois. So we watch them as a vector species, even though they currently don't spread disease here. Now, I mentioned West Nile virus. West Nile virus is a virus that is spread in Illinois primarily by the Culex mosquitoes. It was introduced to the US maybe in 1998, 1999, found first in New York City. Here in the red, those are the states that found it in 1999. Green is 2000. Blue, 2001, it had gotten to Illinois. And by 2002, it had gotten to the West Coast. It seriously spread that quickly across the US. Now, West Nile virus is a bird mosquito cycling virus. So the mosquitoes bite the birds and get infected by the birds. They then go on to bite other birds and they infect those birds, which then infect more mosquitoes, and it goes back and forth. Humans and dogs and horses are all dead-end hosts. We don't infect mosquitoes. We don't get enough virus in our system to infect mosquitoes, but we can get sick. And if you look at this middle map, this is where cases occur in the US, and this is accumulative over a period of time. And you see, California is a big risk, but this little blob right here, that's the Chicago area. Chicago, as some of you may know, was built on a swamp that was filled in. Well, the whole state of Illinois was built on a swamp that was drained and filled in. That's really great for Culex mosquitoes that are floodwater that like areas where water comes in and then goes out again. So the Culex mosquitoes live really well here. And something about the Chicago area is really great for the maintenance of West Nile virus in the population. And you'll see here on the right, this is for the entire US, the incidence of West Nile virus over the years. And you'll see we have low years, we have really high years. 2012 was a bad year, but it just varies year to year. And it depends on a lot of different things, what will make a bad year versus a good year. So this is West Nile virus. Now let's switch to our other vector of interest in Illinois, and that's the tick. Now, ticks, they might have slightly different what we call phenology, which is when they're active at different stages, but they are a far more complicated life cycle than the mosquito. You see, ticks will lay their eggs sometime in late spring. Those eggs will hatch into larvae during the summer, and those larvae have to find a host and take a blood meal. And then they fall off that host and they overwinter, and in their second year, then they molt into nymphs. Those nymphs then have to find a host and take a blood meal. And those, after taking a blood meal, those nymphs fall off and overwinter. Some of them will become adults in the fall. Others will overwinter and become adults in the spring. And those adults then have to find a host and take a blood meal, and then they will lay eggs. So you have, over the life of a tick, 3 different blood meals from egg to egg. This is a problem, because remember how I said with mosquitoes, they're spreading West Nile virus by biting a bird and then getting infected and then biting another bird. And not all mosquitoes are going to take these multiple blood meals during their life. So it's not as much of an issue that they're biting multiple times. They're not doing it all the time. Ticks have to bite multiple hosts in order to survive and get to the next stage of life. That means that these larvae could be biting an infected host, and the next time when they're a nymph, they're now infected and they have to bite somebody else. And so they could be passing on whatever they picked up when they bit as a larvae. And those adults, again, they've had 2 opportunities to get infected, and now they can spread it as well. So the ticks with this multiple host life cycle are really well set up for transmitting disease. We have 4 vector ticks of concern in Illinois. The 3 biggest that we talk about are the dog tick, or Dermacentor variabilis, the deer tick, or Ixodes scapularis. This is the one that most of you have heard about. It's also called the black-legged tick. You see the black legs there. And the Lone Star tick, or Amblyomma americanum, and this is why we call it the Lone Star tick, because the females have this beautiful white spot in the middle of their back. There is also the Gulf Coast tick, Amblyomma maculatum, which only in the last couple of years we've established that it is present in Illinois and it is breeding in Illinois. So these are 4 different tick species. They carry different diseases. They have different preferences for hosts, for habitat. So we have to consider all of these when we're talking about tick-borne disease. And what diseases do they carry in Illinois? There's actually a wide range. Everybody pretty much has heard of Lyme disease, right? So Lyme disease is the most commonly known, but actually in Illinois, we see a wide range of different diseases. So all of these in green, spotted fever group rickettsiosis, which includes Rocky Mountain spotted fever, tularemia, anaplasmosis, and ehrlichiosis. These are all bacterial diseases. Bourbon virus and Heartland virus have been found in Illinois, and those are viral diseases. There are some other viral diseases, but they're less common, and even these are quite uncommon. And then in brown, the one that almost nobody has heard of, but it does strike fear into the hearts of many, which is alpha-gal. It's not actually a pathogen, it's an allergic reaction. So something in the saliva of the Lone Star tick actually creates an allergic reaction, which then creates an allergy to red meat. And so for some people, not everybody, who have been bitten by a Lone Star tick, and we're still learning how it happens, they become allergic to red meat. Sometimes deathly allergic to red meat, sometimes mildly, and sometimes it's only for a year or so, and sometimes it's for life. So the alpha-gal, we're still trying to understand it. It's a very interesting phenomenon, but it definitely is something that we see in Illinois and we hear about in Illinois, and it's not very well known, which is unfortunate because people care about being able to eat red meat. So when we're talking about vectors, we have to talk about climate and weather and landscape before we can talk about prediction. And climate, I'll just say, is not the same thing as weather. Climate means long-term trends and long-term behavior of across the whole year over multiple years. So we see here in these maps between 1970 and 2016, the average minimum January temperatures in the US, if you look at Illinois, it has changed dramatically over that period of time. Why does this matter for us for vectors? January temperatures are sometimes used for prediction because they're a really good sense of how severe is the winter. If you have a severe winter, you get fewer vectors overwintering. Because remember, they have to hide up for the winter. They're not out seeking if the winter is cold enough. They're trying to hide and maintain themselves up until the next year. Well, if the winter doesn't get as cold, not only do you have more surviving through that winter, the more can last, we actually now are hearing about ticks questing in January in Illinois. So because of this climatic change, this climatic shift that we're getting warmer winters, we are actually seeing more vectors surviving the winter, which means more vectors active in the summer. And we're actually seeing vector activity in the middle of winter, which is not good as far as preventing disease, that we don't have a down season anymore. So let's go back to West Nile virus and talk about these mosquitoes. So some colleagues of ours have done some really wonderful work looking at what temperature does to mosquitoes. So people think global warming, that means mosquitoes are going to be everywhere. Not exactly, because you see, as the temperature increases, you see here on these x-axes, the biting rates will increase, but you get to a certain point and the mosquitoes stop biting because they say it's too hot. Same thing as far as survival over time, or their development rate, how quickly they grow to an adult, or their lifespan. They tend to grow as the temperature increases, but if the temperature gets too hot, it actually drops off. So you could see areas of the US in which mosquito activity will actually go down with hotter temperatures because the mosquitoes just don't do as well. And what does that mean for West Nile virus? Well, they've used this to compare what they think the R0, or the basic reproduction rate of West Nile virus, would be at different temperatures. So this is an idea of how many people or animals a mosquito is going to be able to infect if it is infected itself. And you'll see that it peaks at about 25 Celsius. So as we change what our temperature scales are going to be, we're going to be moving to different places on this curve, which means that we will see different risk in different parts of Illinois as we're seeing shifts in the climate. Now, as far as the Aedes albopictus, this tiger mosquito that I talked about, we have seen, and this is from some of our own work that we did with our statistical department here, we looked at over time where these mosquitoes have been found. I will say, and a caveat for this, they're not always looked for. So here, if black means that we don't expect it to be there, that could be just because we don't expect people to be looking for it. But if we tried to control for that to some extent, and we saw where we would predict these mosquitoes to be found over time, and you'll see that we're moving as we go from 1986 up to 2015, our probability of having these mosquitoes present has been moving north and central. And we do know that they are well established in the Peoria area. I know, to my detriment, that they are well established in Champaign-Urbana. I do see them in my yard on a regular basis. So we know that they've moved up to central Illinois, and we have found them in the Chicago area. So the Aedes albopictus is very sensitive to this overwintering, but as we're seeing the overwintering temperatures increasing, we're seeing more of the mosquitoes being able to survive. Now, what about ticks? There are a number of different models from different groups here on the right-hand side, looking at predicting. Here's with the Gulf Coast tick, here's with the Lone Star tick, here's the black-legged tick, looking at expansion over time, especially given the changing in climate. As a mosquito, just like mosquitoes, ticks are susceptible to the changes in climate. Well, what have we seen in Illinois? And this is some work that a collaborator lab has done, looking at when we first detected the different ticks. So this is Lone Star, the dog tick in the middle, and the black-legged tick on the right. And you'll see that over time, we're finding the ticks in more and more places. The Lone Star tick, we started finding a little bit later, really mostly in the 2000s, and finding more and more places over time. The black-legged tick here on the right, we saw a lot of it in the 90s, but during the 2000s, it started to move into more and more areas, especially this agricultural land, which in general, people who study black-legged ticks say, well, you know, you won't see those in agricultural land, but we're seeing them move into these areas. And then the dog tick has been pretty present in the state throughout, but we are seeing it expand, or maybe we're just finding it in more places. But we definitely know that the Lone Star tick and the black-legged tick are both expanding into Illinois. And if you look at this map of the Gulf Coast tick, you'll see that they didn't predict it would be in Illinois. Well, now we know we've found it all the way up into central Illinois, including in the in the in the backyard of one of our team members. So we know that it's moving up into central Illinois beyond where it was even predicted to be. And we suspect that it has something to do with climate. So that was all climate, thinking about long-term trends. What about weather? Weather is really important for vector-borne diseases because vectors are small, and so they're susceptible to slight changes in precipitation, in temperature. You remember those graphs of what a change in temperature will do to all of the mosquito development and biting rates. Now, here is for Springfield, Illinois, throughout the year, what our normal precipitation and temperature changes are. And you can see, precipitation is pretty active in the spring and through summer and then drops off in the fall. We've got temperature going up. We've got day length changes. Day length makes a big difference for West Nile virus because of the birds and what the birds are doing. So for West Nile virus, if you remember that graph, that pretty well tracks with this graph of when we see human cases of West Nile virus. And this blue line here is the mosquito infection rate, which is an indication of how infected our mosquitoes are. And you can see that the mosquito infection and the human cases track pretty well, and they follow pretty well this precipitation and temperature combination. And that's because of this life cycle over here, that they need the water and they need the warmth to be active and to be out and biting. So we'll see sometime in early July, we'll start to see activity in the mosquitoes. And then shortly after, we'll start to see cases pop up in people. With ticks, it's a little bit more complicated. So ticks dry out, what we call desiccate, pretty quickly. So they have to be very careful about not getting in too hot, dry temperatures. But they also can't be out searching for hosts when it's raining because the rain is big compared to them. So they're going to be looking at times when it's very humid, which means that when we try to find a connection between when we find different species and life stages, it's related to the time of year, spring versus summer. Sometimes some prefer spring, some prefer summer, some prefer fall. The region, whether it's central or southern Illinois, makes a difference because that weather changes between those areas, but also the precipitation, the temperature, and vapor pressure is how we're measuring humidity because we don't have humidity measures down at the tick level. So we're using vapor pressure. And what we see is that they like the warm, but not too hot, humid weather that's not actively raining. And that's pretty consistent throughout. And that matches what we know about tick biology, that they're going to be out when it's humid and it's not actively raining, but maybe it's recently rained, which is exactly when the hosts are going to be out. So land cover is another thing we have to think about for prediction. This is the land cover of the state of Illinois. If in case you're wondering, this light brown area, this is corn and soybeans. So this is all the agricultural land, which is really amazing to look at. But then you've got the green bits, these are the forests, and the pink bits, these are the urban landscapes. And you can see that there's quite a variety in Illinois, and it varies within counties as well as across the state. So what do mosquitoes need? Mosquitoes need water because they need somewhere to lay their eggs, and they need green space. For one thing, the Culex mosquitoes want to bite birds. So they want areas that birds are in, birds like trees and bushes and other types of cover. So the Culex mosquitoes are going to be around the areas where birds are going to be, but also that has water. So this is one neighborhood in the Chicago area that our team mapped very closely where the green space was, where the trees were, where all of the water sources were, and looked at how many Culex mosquitoes you could collect in a night in the different areas of this. And you'll see that there are much bigger circles, which means more Culex collected in these green areas that have some form of standing water near them. They like to be around wet green spaces. Ticks need hosts. So remember I said the ticks need to have 3 different blood meals to go from egg to egg. So they want hosts. They like these white-footed mice. You'll hear about possums and whether or not possums are great at grooming off ticks. It's not necessarily true. There's a lot of debate about that right now. But you'll also see foxes, all of these, what we call the mesocarnivores, the middle-sized carnivores. Ticks will be happy to host on them. And of course, as many of you have heard about, white-tailed deer, great tick hosts. But it depends on the life stage, actually. So the larvae will really like these small white-footed mice, which is not good for us because if their first meal is from a white-footed mouse, nymphs also like the white-footed mice, and the white-footed mice are really good at carrying the pathogens that we're concerned about, particularly the one that causes Lyme disease. So if we've got larvae and nymph all biting on the white-footed mice, they're really at risk to get infected. Deer are actually not good at transmitting Lyme disease. They're not bad, they're just not good. What deer are is a really good blood meal for a lot of adults. So the white-tailed deer are giving a lot of adults a good place to get a good blood meal so that they can go then lay a bunch of eggs. And the white-footed mice are a really good place for the younger life stages to get infected. So we have all of these hosts. Well, where do the hosts live? The hosts live in the forest edges or in the forest. But some of these hosts, like the white-footed mice that we're really worried about, they do really good living in these forest edges, in the little bits of forest within a suburban development. Same with white-tailed deer, which are really good at feeding the adults, that they like to live anywhere that there's a little bit of trees, which is not good for us in Illinois, where we have a lot of interrupted bits of forest, because that's a good place for these really good hosts for ticks and tick-borne disease to live. So I said that this talk was about prediction and prevention, and I haven't really talked about prediction yet. Our team has done a lot of work on trying to predict where West Nile virus disease will be. And we actually have a website. You can go to it, PresVBD. It will predict by county where West Nile virus activity in mosquitoes is going to be. So if you want, you can go look up your county and it will show you for this year, based on the weather and the weather forecast, what we think the next week is going to be like as far as mosquito infection. And this is all good. This is great that we have this service out there that you can go look at the predictions. But, well, when we asked the people who do mosquito control, at what spatial and temporal scale do you actually make your decisions? So here on the x-axis, we have spatial scales from the county level, which is what that map is, down to the point level, which is the individual points, like an individual house. And this temporal scale from the multi-year up to the hourly. And some of their decisions are made at the county and township level and at annual level. So predicting how much larvicide do we need to order for the year, and how much should we be planning for a bad year or a good year. But a lot of their day-to-day decisions are made at the weekly and daily basis, and they're working at smaller operational levels. So operational levels being local, streets, blocks, they can't do everything everywhere all at the same time. So they have to decide, which neighborhood am I going to go into and treat for mosquitoes right now? Well, when we took that same scale and we mopped out not just our prediction models, but all of the West Nile virus prediction models that exist out there, you'll notice there is nothing predicting up here in the daily to hourly scale that they're making a lot of their decisions on. So we're not actually producing the prediction models that will work at the scale that the people who are doing mosquito control actually need. And you might say, well, get on that. What are you doing? And the problem is data. And here we have a map for a 3-week period in 2016, which was a very bad West Nile virus year, where all of the traps, mosquito traps in Illinois were. And you'll see the Chicago area is just littered with mosquito traps. We've got lots up there. And there are other counties that are doing a really great job. But then there are some places that just have no data. And the problem is that in order to build the models that will predict up here in this area where they really want to make these decisions about where should we put the larvicide and where should we spray, we need data at this level. And we don't have it in much of the state. And Illinois is pretty good as far as mosquito surveillance. So there are other states that are even worse. And I know I have compared our model for predicting human disease to a model that is just based on where human disease has been in the past. And the model that just says, okay, we're just going to go based on what we've seen in the past is actually much better at the level of the state or at the county level across the state, just because we don't have the data that we need for our model, which again is not even at the fine point scale that the people who do the mosquito response need. So we need a lot more research on how to actually build these models that are at the fine scale that we need for decision making, but that don't rely on having huge amounts of data. So to be continued on where we are going with that, but that's where my lab is working right now, is trying to figure out how best to make these predictions using the data that we actually have. So that was on the mosquito side. On the tick side, we simply don't have the data in the state of Illinois yet to be able to make these predictions. And we have been doing our best to increase surveillance across the state. So these maps are from a publication we have describing the first year of iTick, the Illinois Tick Inventory Collection Network, which is our community science program, where we ask people to submit ticks that they find on themselves or their animals, and let us know about where they're finding it, what they were doing when they found it. Now we've partnered with the Tick app, so moving into the future, you can actually just log your tick find on the app, and we'll tell you what kind of tick it is, and then we know where you were and what you were doing when you found it. This helps us know where the ticks are much better. But the colors on here are what we had established historically as where we knew the ticks were. And these cross hatchings are all of the additional knowledge we have with just doing more surveillance, more tick dragging, which is where we're going out and actively looking for ticks, but also partnering with community members and asking them to submit ticks to us that they've found on themselves. So for ticks, we just really need to increase our surveillance across the state. And once we've built some of that data, we can start making these prediction models. Now, how about prevention? Because I did say I was going to talk about prevention. Well, with the mosquitoes, some of the most recent work we've been doing is looking at, with our partners up in the Chicago area, they spray for adult mosquitoes on a fairly regular basis during the summer whenever they get nuisance complaints or when they see a lot of West Nile virus activity in the mosquitoes. And so they asked us to tell them, what does their spray actually do to the mosquitoes and their infection rate? So spoiler alert, doesn't do anything to the infection rate. So which makes sense. Spraying for mosquitoes isn't going to change which mosquitoes are infected. They're all susceptible to the spray pretty much equally. But what we find with the number of mosquitoes out is that for 1 to 5 days after the spray, we see on the most part, and each of these circles is a trap, is an individual mosquito trap location, we see big decreases in the number of mosquitoes we're trapping for 5 days after a spray. But once you hit a week after the spray, it's not just back to normal, it's actually increased from normal, which is actually, it might sound counterintuitive, but it's actually what the people on the ground had reported to us that they think happens, is that we see a bounce back. We spray, we knock down all the mosquitoes, and then all of the larvae hatch and we get more mosquitoes than we had before we sprayed. And that increase actually lasts for 1 to 2 weeks after the spray. So the sprays are very helpful for 5 days, just to knock down the number of mosquitoes in the area, and then they bounce back. And there are other people who are now working on, well, what if we sprayed twice in a week? What if we sprayed 3 times in a week? What would that do? And also thinking about what other things can we do to bring those mosquito numbers down. The other thing that we work on in our lab as far as prevention is doing outreach. Especially with ticks, as I said, we don't have the data yet to do a lot of the prediction, but we do know where ticks are likely to be, and we do know how ticks behave and where people get exposed. So we do a lot of outreach. This is a infographic we made specifically for hunters. We know that hunters are getting exposed to ticks when they're out hunting, also when they're out looking for hunting spots. So we tell them, okay, here's how you check for ticks, here's how you prevent them. You can treat your gear, you can use repellents. Here's how you report them to us if you want to know what kind of tick you have, and also how to protect your dog. And what we hear from hunters that we talk to is they care a lot more about protecting their dogs from ticks than themselves in most cases. So hopefully if they can check their dogs for ticks, they'll remind them to check themselves for ticks as well. So a lot of our work we do is just outreach, things like this talk, coming to talk to people about where the vectors are in Illinois and how they can protect themselves. So hopefully people will protect themselves more, and then eventually we'll figure out how to control these vectors better, and we'll be able to make the rest of Illinois safe. Thank you for letting me come talk. I have to thank everybody who I work with on all of this work, particularly the Midwest Center of Excellence for Vector-Borne Disease, which is a CDC-funded center, who have been wonderful at developing our knowledge of vector-borne disease in the upper Midwest. Also, I have funding from the Department of Defense on tick and tick-borne disease prediction, which we are working on. But all of the members of my lab, so the long-term members and the summer help, we get a big team over the summer. The Illinois Natural History Survey has been a big help, especially Nora Mateus-Pinilla in the Wildlife Epidemiology Lab and the Medical Entomology Lab. The Department of Statistics helps us because these prediction models take a lot of statistical expertise. Former members of the lab, Surendra Karki, did a lot of the initial work in the Chicago area. And of course, our mosquito partners on the ground, and they're also tick partners as well. They send us ticks. The Northwest, North Shore, and Wheaton mosquito abatement districts are huge helpers. And we also work a lot with the National Center for Supercomputing Applications, developing these tools, such as the PresVBD website, to get the word out. And big thanks to Marilyn O'Hara Ruiz, who was my colleague who introduced me to vectors and who taught me all about them. And we lost her too soon just a few years ago, but she really built this program, and we're just trying to keep it going for her. So thank you all for letting me come talk, and I hope there will be questions and interest in the future. Thank you.